Our next speaker is going to be Alex Eng. So as I said, Alex was part of the UCSF iGEM team last year, which was primarily made up of students from Lincoln High School, one of the public high schools in San Francisco. And he's going to tell us about the UCSF project, which was trying to design a new cellular organelle.
All right. Hello, everybody. As you just heard, my name is Alex Eng, and I participated on the UCSF iGEM team in 2007. And today what I'm going to talk to you guys about is a formation between UCSF and Lincoln High School and what that was all about, and also our project from last year, which was to build a synthetic organelle. And after all that, I'm going to talk to you a little bit about this year's team. Now, University of California at San Francisco, or UCSF, is a great school, but it doesn't have its own undergraduate program. Now, because of this, it was always kind of hard for UCSF to make its own iGEM team. So what UCSF has traditionally done in the past was to look around at various high schools to look for team members. But for 2007, they decided to change things a bit, and what they did was they went to one specific high school, Abraham Lincoln High School in San Francisco, to obtain most of their team members. Now, Abraham Lincoln High School is one of many public schools in San Francisco, and it's where I went to school, and it's how I got introduced to iGEM. Now, why did UCSF decide to pick Lincoln High School? And the answer to that is that Lincoln has this very unique two-year biotechnology program that's taught by two great professors or teachers, George Kachianis and Julie Reese. Now, in the first year of this program, students learn about basic biotechnology. They learn about things like what it is and how people use it and what techniques they use to do it. And a very cool thing about this class is that it's very hands-on. And what I mean by that is that it's very lab-based in that the students in the first year program learn how to do things like pipette or run gels and set up PCRs and other cool stuff like that. So in this first year of the program, there were probably about 100 or so students who participate in it. And of those 100 students, only 25 to 30 students get selected to continue on to the second year of the program. Now, in the second year of the program, students go more in depth and learn about things like how to design plasmids, and they even run a Western blot, which is really great because not many students in high school get the chance to do these kinds of techniques. For instance, I'm a sophomore undergrad in college right now, and I haven't even been able to pick up a micropipette in class yet. So that just goes to show how much experience these kids have and how much they learn from this class, which is why it was a great place to look for students for the iGEM team. So the way I was introduced to iGEM was that one day my teacher, George Kachianis, asked a few students to stay after class. Now, when I first heard this, I wasn't sure what to think about it because usually when a teacher asks you to stay after class, you're usually in trouble for something. So luckily I wasn't in trouble. I stayed after class, and he told us about this iGEM competition. And at first we didn't know what it was, but he told us that it was going to be a challenge because it's mainly an undergraduate competition, and we were just high school students. But we weren't thinking to win. We were just wanted to the experience of this, being able to work in a lab. So we were just like, yeah, let's do it. So that's how we got introduced to the whole iGEM. So every week, once a week, we had a meeting after school to give us a basic introduction to what synthetic biology is and what it's about. Now, of the students in the second year of the program, only 5 were selected to participate in the UCSF iGEM team from last year. Now, along with those students, we also had one student from Palo Alto High School and one senior undergrad from UC Berkeley join us as well. Now, the way our team was structured was that each high school student was paired with a mentor from the lab. And these mentors were either graduate students or postdocs from the lab. And from my perspective, it was great because coming out of high school, we all had this general understanding and general knowledge of biology. But for iGEM and for our project, we needed a more in-depth knowledge of specific systems. So these mentors were there to give us the basic information and basic background we needed in order to understand what we were dealing with. And along with that, they were also there to answer any questions that we had whenever we had any snags. So even though we each got individual attention, at the same time we were very independent in that we did our own experiments and did our own data analysis. It was only when we had a snag or we hit a snag or had a question that they would help us out, and after that we would go back to doing our own thing again. So that was our team from last summer, but our project was to build a synthetic organelle. Now, what's so cool about this, right? Well, in biology, cells use membrane-bound compartments to make specific biological processes more efficient. So, for example, the mitochondria and its energy production. It's efficient at what it does because the compartment allows the mitochondria to localize proteins together and have them interact more, which makes them more efficient. A second reason why biology has these compartments is to limit toxicity in the cell. For example, the lysosome. And the lysosome is filled with toxins and enzymes that would probably harm the cell if it were released into the cell. But because these enzymes and stuff are contained within these membrane-bound compartments or organelles, it doesn't hurt the cells. So what would we do with our new synthetic organelle if we made one, right? This new organelle would be potentially useful because it could, house any kind of system that anybody would want to put in. For instance, you could put a biofuel factory in, or a drug factory, or anything. And again, this would be great because it would make that system more efficient and also limit toxicity by containing potential byproducts or toxins that your system might release. Now, so the steps in building new organelle is that we would first start with a wild-type yeast cell and then introduce our new synthesome, and then eventually people will be able to use it and put their systems in it. But for iGEM and for the summer, we decided to concentrate just on this first step, which was to create a spatially distinct membrane using a new molecular identity. So what do I mean by molecular identity, right? And so the first question we had to ask ourselves was, how does nature naturally differentiate organelles in the cell? And the answer to that is that nature tags different organelles using these things called phosphoinositides. Now, these phosphoinositides can be phosphorylated in 3 different positions, the 3, 4, and 5 positions. And being phosphorylated in a different combination of these positions resulted in a unique molecular identity that was specific to a specific organelle. So, for example, not being phosphorylated in any of these positions was unique to the endoplasmic reticulum. Now, I have some more examples for you. If you look on the right side of the screen here, this is a diagram of a cell and a bunch of organelles. But if you look more closely, each organelle and each membrane compartment is color-coded to the molecular identity using these phosphoinositides. So, for example, being phosphorylated in the 3 and 5 position was unique to the late endosome, and being phosphorylated in the 4 and 5 position was unique to the plasma membrane, and being phosphorylated in the 3 position was unique to the early endosome. So basically what I'm trying to say is that each organelle in the cell has its own unique molecular identity that was specific to itself. So with the possibility of being phosphorylated in 3 different positions, it meant that there were a total of 7 different combinations of these phosphoinositides. And in higher eukaryotes, all 7 of these structures are used. But in yeast, which is what we used for last summer's project, the phosphoinositide 5P, or 5-phosphate, or PI5P for short, is not used. So our goal for the summer was to create a new compartment using this novel phospholipid structure, the PI5P. So that's how we would differentiate our new organelle. But how do we go about making this new complex, and where would we get our new membrane from, right? So what we decided to do was take advantage of nature and what happens to the mating receptor and its endocytosis pathway. So what normally occurs is the mating receptor gets stimulated by a mating pheromone or mating factor, and that triggers endocytosis. So it goes to the early endosome and then goes to the late endosome, where it has the molecular identity of a PI35P. That is then recognized by the vacuole, and it merges with the vacuole. So what we had to ask ourselves next was, how do we make our PI5P? Now, if you look at this late endosome, it's the pi3-5p complex, but what we wanted was a pi5p, which is very close. So the next place we looked to was higher eukaryotes, because we knew higher eukaryotes had this molecular identity and use this molecular identity. So what we found was that mammalians and higher eukaryotes have this lipid phosphatase that would take off the phosphate at the 3' site and make it our new pi5p. So what we wanted to do was introduce this lipid phosphatase and recruit it to our receptor, and that would essentially hijack our late endosome and make it our new cell compartment using this novel phospholipid. So that was our plan, and that's what we did over the summer. But to sum it all up, because I know it's a lot, what we wanted to do was introduce a mammalian lipid phosphatase that makes this new pi5p complex. And this phosphatase would target a specific subpopulation of vesicles to make our new compartment. Now, we did this over the summer, so let's go look at our results now. So for purposes of seeing our new organelle, we tagged our mating receptor with GFP. In this picture that you see here, the left side is the control without the phosphatase, and the right side is our experiment with the phosphatase. So this picture is the picture of our yeast before mating pheromone stimulation. So you can see that the green is on the outer part of the cell, which is great because that's where it's supposed to be, because it shows that our mating receptor is on the cell membrane. So after mating pheromone stimulation, if you look at the control side, you can see some dots which show that our mating receptor is getting endocytosed. But if you compare that to the right side, you can clearly see that there are more green dots, which not only show that our mating receptor is getting endocytosed, but it's not merging with the vacuole. So these preliminary results suggest that we have made a new cell compartment. And this data looks so promising that we actually have a postdoc coming in to work and continue on this project. Now, we took all our results and all our information and all our ideas, and we brought it to iGEM, and things went great because, as you heard earlier, we were finalists. We did so great, in fact, that we were featured in our local newspaper, the San Francisco Chronicle. And our biotech class from high school is also featured in the New York Times, which is pretty big. So that was cool. So for this year, this year's theme for our project is chromatin-based gene silencing, and that's all I'm going to tell you guys. But to work on that project, UCSF decided to take 6 new students from the class at Lincoln High School. Along with those 6 students, UCSF asked 2 former students from last year's team to come back to this year's team, and I am one of those students, as you can see right there. But something different about this year for these 2 students and for me was that we had a different role on the team. Last year, our role on the team was to be students, and we were paired with a mentor, and where we asked questions, and we just soaked everything in and learned as much as we can. But this year, UCSF switched our role in that they asked us to be mentors to the new incoming high school student. So I was paired with a high school student of my own, and he would ask me questions whenever he needed help, and I'd try my best to answer him. And so that really tested my knowledge of what I thought I knew, and I think that was a good way for me to learn as well. So I think that UCSF threw this challenge at us, and I think we handled it pretty well. Now, something else UCSF decided to do different this year was invite international students to come work on our team this year. Now, the Jamboree is fun, and it's great because we get to meet people from around the world, and not only around the nation, but from around the world. And it's great, but these connections only last for 2 or 3 days. You know, the iGEM competition is only for a weekend. So UCSF decided to continue these relations that we made by inviting 3 students to come and work on our team for this summer. 2 of these students are from Peking University, and one of these students are from University of Ljubljana. The 2 students from Peking University are Dan and Lingli. Dan is going to be a grad student next year at UCSF, and Lingli served as a mentor to one of the high school students this summer. Andre is our 3rd member, and he is from the University of Ljubljana. He's an undergraduate there, and he helped out with our project as well, and also served, or also had a mentor role as well. Now, not only did we take students in to come work with us over the summer, but I guess you would say we exported one of our students out. Eric Meltzer, who is from last year's iGEM team, the 2007 iGEM team, went to Peking University this summer to work on their team for this year. Now, I think this international program is great because it brings students and people from around the world and brings them into one lab and one room, and they get to share ideas. And that's great because that's what science is about. And science is about collaborating and sharing information and working together. Louis Pasteur once said that science knows no country because science or knowledge belongs to humanity and is a torch which illuminates the world. So this program really plays or promotes the idea that science should be shared by everybody. So my overall experiences from the last 2 summers was great because one of what I talked to you about already is the student-mentor relationship that I had. You know, the first year I get to learn so much, and this year I got to learn so much also, but this year they added a new dimension to what science is about, and it's about teaching people under me. So hopefully that can continue. And also, what I talked about earlier, or what I just talked about, was the International Exchange Program, which brought students from all over the place and bring them to one room where we got to collaborate. So that was great as well. And another thing is that we got to go through the whole scientific process in that we got to plan our experiments and do our experiments and also troubleshoot and see our own results. You know, in high school, students are given protocols and the experiments are expected to work no matter how much you screw up. But for the summer, you know, that wasn't really the case. And so we really got this experience and it was great for us. So I hope programs like this and the iGEM competition can continue so that students like me and other people around the world can get the same experiences and same opportunities that I did over the summer. Thank you.
Okay, thank you very much, Alex. Do we have questions? Pam? Speak loud.
First of all, that was fantastic.
Why don't you give it a mic?
Okay.
Many faculty interviews. I was curious, so you're a sophomore in college now, you said? So I was curious how you've translated this into your college experience. You made sort of a remark about not having the same opportunities, and so I'm wondering, do you feel like you're getting any opportunities in your college experience to do similar things, like the interview?
Yeah. So going to class right now is really boring because I'm going to San Jose State right now. So yeah, like I said, going to class right now is really boring. It's easier for me to follow things now because of this program, because of the things I learned. And also, I have applied for a lab position at my school, and I've gotten a letter of recommendation from UCSF, and I've gone through the interview process, and now I'm just waiting to hear back. Something you don't know is that Robert Ovadia, who's from last year's team, is now on the Caltech team this year. So things are really going great for us.
It's hard to keep these kids away. Alex still comes in every Friday. He comes back from San Jose and comes into the lab every Friday. So.
I think it's important to think about how to make the college experience better.
Yeah. Good point. Other questions? Julius.
Do you have a sense of how much of the phosphatidylinositol label you need to change before it's no longer recognized by the uptake machinery, like percentage-wise per compartment? No is fine.
No.
Thanks.
Other questions? Right here.
So when you were in high school, what was the criteria for like first-year students to move on to the second year? Was there some sort of test or?
There was no test. I think students just had to show interest, and a lot of people showed interest. I think people kind of just got lucky to get into the class, and I was one of those lucky people because as this program continues in high school, it gains popularity, and a lot of kids really want to do it now, and they hear about this opportunity that they get, and so space is really limited now, and I think they're trying to expand this program.
Okay, thank you.
Yes, over here.
understand each of your high school students was paired with a postdoc or a researcher mentor. I wonder if you had a sense of whether there's a difference in the mentor-mentee relationship with that relationship and your high school students compared to some of the other undergrad teams and their mentors.
I'm sorry, what was the question again?
It seems like a pretty intensive mentorship working with the high school team, and I didn't know whether you had a sense of there was more hands-on assistance or how much the students did versus how much the mentors helped them with, and whether that was the same kind of interaction that the undergrad teams had with their mentors. Was there the same degree of researcher support with the team members in your high school case as compared to the undergrad case?
Well, I don't know how much mentorship that the undergraduate teams got, but for us, we kind of went through this boot camp thing in the beginning of the summer where, they kind of held our hands a bit. But as the summer went on, we got used to lab and we got used to working, and, they kind of just let us go and do our own thing. So, yeah. Yes.
So now that you have a new compartment, how do you envision to get things in and out of your compartment?
So we can target things to the receptor, and when things get endocytosed, it'll be on the inside. I think there are other ways, but those are things that we haven't figured out yet because we haven't got the synthisome working yet. So first step first.
Any other questions? Okay, just before we end this, I want to point out that we're also very lucky to have George Kachianis here at the meeting. He's the biotech teacher at Lincoln High School who runs this 2-year program. George is here somewhere, but he's going to be speaking on Sunday morning in the C session. So those of you who are interested in learning more about the program that he's set up, this really model program, please go to that session. Okay, thank you very much to both Alex and to the team from Beida.